Fahim comes with a lot of experience in this field. We're happy to have them with us, but just before we start, I'd like to make a couple of housekeeping remarks. Questions can be sent through the chat button, and all questions will be addressed to the host. And the second remark is that everyone is going to be muted throughout the session. And with that, I welcome Dr. Fahim to the talk. Thank you.
Thank you, Naguib. Can everyone hear me? Naguib, can you hear me?
I can, yes.
Perfect. Thank you. Thank you, Father, for your prayers. I would like to just tell you the reason why we thought this session would be of interest to everybody. And that is because in the past few weeks or so, as vaccines for COVID were approaching licensing, there appeared to be plenty of anti-vaccine misinformation, which may hurt wide vaccine adoption. And as a vaccinologist, this actually is disheartening to me because I know that these vaccines are safe and reliable and will actually bring the COVID epidemic to an end. The reason why that is important, why everybody is free to either choose to be vaccinated or not, we have to realize in order for us to get what's called herd immunity, we need to immunize over 60% of the population. So over 60% have to be immunized and protected against COVID-19 so that we can stop the epidemic. And that's the reason why we need to all work hard at convincing others to get the vaccine. So my objective for this talk is to make you better informed as an individual and convince you to take the vaccine if you are hesitant. I'm happy to answer any question you have, but I'm also hoping that you would convince others in your community. With that, let me just tell you how we're going to divide this talk. While I'm not going to get into a very detailed scientific information, I feel it's necessary that we talk about some basic aspects about the virus, about our own body and how it forms proteins from genes, from the genetic code that we have, and then also how our body fights viruses, how our immunity system or immune system works. We'll also talk about the types of vaccine that you may see in the next few months after they are licensed. We will talk about how safe they are. And one of the major questions that I always get is how can companies develop vaccines that fast? I can tell you prior to COVID, the fastest vaccines were developed in somewhere between 6 and 20 years. I developed vaccines, the fastest I've been able to is 8 years. So how can we make it in 11 months? We'll talk about that. We can also talk about some additional misinformation, and then finally, an advice as to whether you take the vaccine or not. So let's start by talking about the virus itself, the COVID virus, and maybe just take a moment to tell you a little bit about viruses and infections for humans in general. We normally, as human beings, get one of two types of infections in general. We get more, but the majority is either bacterial or viral. The difference is huge between a bacteria and a virus. While a bacteria is a living organism, means that it can replicate on its own. It doesn't need anything else to replicate. You just give the bacteria some oxygen and some nutrients, and it can replicate. Viruses, on the other hand, are actually not living things. They cannot replicate on their own. They need to use some other mechanism, some other machinery to get them to replicate. So the viruses, when they enter the human body, they use the machinery of the human body to replicate, unlike bacteria, which can do it on their own. So for example, we have antibacterials, like the antibiotics that you get. we can develop those because they can stop the replication of the bacteria itself without harming the body. On the other hand, if we want to do that for the viruses, then we inevitably will actually harm ourselves as well. That's why when we give antivirals, we are very specific about how to use them and where to use them and when to use them. With that in mind, which would be important later on in the discussion, I just want to introduce the COVID virus itself. As you can see, this is, I'm sure all of you have seen this diagram before. It's, this is the shape of the virus itself. And on top here, you can see these spikes. That's why we call them a spike protein. These are the very important parts of the virus that allows it to enter the human cells. And I will talk about that in a second. These spikes are really what we in the vaccine field are trying to develop vaccine against, this spike specifically, and I'll show you why in a second. So some important facts and characteristics of the COVID or the COV-2 virus. First of all, by the way, COVID-19 is the actual disease, but the virus we call it SARS-CoV-2 because there was original SARS, you may recall, in 2004, 2005. So now this is almost the same, that's why we call it CoV-2. So the virus itself called SARS-CoV-2, the disease is COVID-19. Okay, so we call it a very smart virus. And the reason why we call it a smart virus is that even before you develop symptoms, if you have the virus, you become infectious a couple of days before you show any symptoms. During that time, you don't know it. You're roaming around, talking to people, congregating with others, infecting them all the time. And then a couple of days later, you realize you're coughing, you have fever and whatnot. You go get tested and you have the COVID-19 disease. Now, that is why it's a very deadly form of the virus because it evades detection until it spreads itself among others before you know it. For example, the Ebola, which is much, much more deadly than the COVID or the coronavirus, way more deadly. Almost 80% of people get Ebola die. However, it is so easy to see that somebody has Ebola, so you isolate them. And therefore, because you isolate them, they don't infect others. They may die, but they have not infected too many people. That's why the pandemic, the epidemic of the Ebola was controlled very quickly in Africa before it spread around the world. The second important characteristic of the virus that will be the reason why we call it a smart virus, it's able to evade the immune system. It's able to get around the immune system. We're not going to get into that, but just to let you know that that is one of the characteristics of it. It causes severe respiratory lower tract respiratory tract, the lungs, infection, but it also infects the upper respiratory tract, and that's the mouth and the nose, and spreads therefore through coughing, sneezing, or breathing. It infects people through the nose, mouth, and eyes, and that's why we ask people to wear masks and put a shield, mostly to protect others from you if you are infected, but also helps protect you from somebody who's infected, although to a lesser extent. One important aspect of the virus that we have recently started to discover in the past few months or so is that the virus actually lingers in the air in small droplets of water or when you cough. And because of that, we say people should not congregate indoors for a long period of time because then the air is stale and people can get infected quickly. So if you're sitting in a community, a small community and whatnot, it's good to open the windows every once in a while. I know it's winter, but I do it here in my own home every once in a while, open the windows to refresh the air so that we don't have stale air that may cause the infection to spread quickly. Additional information, not everyone is symptomatic. We have asymptomatic people, and they would be even more dangerous than the symptomatic ones because they spread the virus without even knowing it until they get tested. We don't know why that is the case. We don't know why some are symptomatic and some are not, but we know that it happens. We also don't know how long the immunity. I'm asked all the time, how long do you think it would be? If I get infected or if I get the vaccine, how long is my immunity? We don't know because it's just been a few months since we discovered the virus. However, we do know that the immunity memory, or the way the body memorizes that it has seen a vaccine or a virus previously, from the SARS-1, CoV-1, which was in 2004, 2005, we were able to test people who got infected with that SARS, up to a few years later, and they still had the memory, which means that the body still remembered what the virus looked like, and therefore had the information to produce antibodies to actually attack the virus if they see it again. We know that the memory is there for a few years. So therefore, we are hoping that for the SARS-CoV-2 that causes the COVID-19, that would also be the case, but we don't know that yet. And one of the things that I'm asked all the time, we hear about mutations. Does that make the vaccine after we take it useless in the future? Something like the flu, where it changes every year or so. And the answer is, we really don't know a lot, but what we know is that the mutations so far have not impacted the spike protein, which is the target of all the vaccines that we have today. I just wanted to share with you some numbers so that you know that would be also important later on in the discussion. The SARS-CoV-2 virus is approximately what we call in measurement 0.1 micrometers or microns. Just to make sure that you understand the relevance of that, this is 10 times smaller than the smallest microchip. And it will become evident why I'm making that comparison as we go on for the discussion when we go to the misinformation. You may have heard the term, or if not, then I'm trying to explain it here, the RO of the virus is 2 to 4. That means that every infected individual can actually infect 2 to 4 people during the period of time where they are infectious. So every person can infect 2 to 4 people. So imagine if somebody is infected and sat in a community, they will infect 2 to 4 people, and those 2 to 4 people then go on to infect 2 to 4 other individuals and so on. So it spreads very quickly. That's the RO. That's not the highest RO. It's very high. The RO, we need it to be under 1 so that we can get rid of the infection. That's why we're saying social distancing, wearing masks and whatnot to reduce that rate of infection. But it's not the smallest. Measles, by example, is approximately 12 to 14. Everybody who has measles can infect 12 to 14 others. But for the measles, we know the symptoms, so we can avoid getting, and you know that if somebody close to you got measles, we don't get people around them a lot so they don't get infected. Here, as I mentioned, the first 2 days or so, you don't know it, so you infect others without knowing that you have the disease. I wanted to share with you some timelines from the time somebody gets exposed to the virus till the time they get to be infectious. First, when somebody encounters somebody who's infected, they will have the virus in their body, but they are not infectious. It stays about 3 days without being infectious. So you get the virus actually in your nose or mouth, and the virus is multiplying, but you are not infectious. That's called the latent period. Doesn't matter what it's called, but that's the period before you get infectious, and that's 3 days. There's also what's called the incubation period before the symptoms develop, and that's 5 days from the first time you encounter the virus. So the difference between those 3 days where you're not infectious and 5 days where you start getting symptoms That's the 2 days here that you are infectious but don't know it. And this means that then this person becomes infectious for about 2 days before they have the symptoms. Individuals would be infectious for about 4 days. So 4 days plus the 3 days of latent period, so that's 7 days that we have now infected. And in a way, you can still be infectious, although not at the high level of infectivity, for about 10 days potentially. So when you add the 10 days to the 3 days, that's why people say quarantine for 14 days to just make sure that you are clearly not infectious. Although it's rarely, or the majority of infections happen in the first 7 days. So it's the 3 days latent period followed by these 4 days of infectivity, high infectivity. That's when you get infective. After that, it goes down dramatically. I'm just going to share with you some basic biology information. Don't mean to get into science a lot, but just briefly because it's important. I know that some are not, don't have a science background, but it's important to know because it will help us understand a little bit of information later on. The DNA, I'm sure you know about, that's genetic code. That's the genetic code that we have in ourselves, bacteria, viruses, everybody has that DNA. The DNA in the body, in the cell, forms what's called RNA. And then the RNA is what makes the protein, and proteins are the building blocks that you and I have. That is the material that you use for everything you do, and these are the proteins. Now, you have to remember that from a DNA you get an RNA, from an RNA you get proteins. You cannot make RNA from a protein. You normally don't make DNA from RNA, although there are ways to do it, but these are very specific things we don't need to get into. In general, you need to know that this is unidirectional, from DNA to RNA to proteins. Okay, with that in mind, I just want to show you how the virus can get into the human cells and how we can protect ourselves against it. So let's assume this is the virus, the SARS-CoV-2 virus. These are the spikes. These spikes are the way the virus fuses or attaches to the human cells. Don't worry about what these receptors are, but enough to know that this spike will then attach to this part of the cell and then internalize and get into the cell and start using the machinery of the cell to replicate. Whether you get infected with the COVID-19 or get a vaccine, the way we fight this virus is produce these antibodies, these small molecules called neutralizing antibodies. That's how we defend ourselves. These neutralizing antibodies bind to this spike protein and prevent it from attaching to the cell and therefore internalizing. So after you get the infection, a few days later, if you're lucky and you recover, you would have produced these antibodies. If you get the vaccine without getting the infection, you still produce these same antibodies, which then protect yourself from it by attaching to the spike protein and prevent it from attaching to the cells, and therefore you don't get infected. So how do COVID vaccines work? As I mentioned in the previous slide, the vaccines contain parts or all of the spike protein or something that can make the spike protein, and we'll talk about that in a second. When injected, these vaccines then provoke an immune response. It actually trains our body to recognize and produce specific antibodies to this spike protein. So in essence, once you see that spike protein, whether it's on the virus or on anything, you see that spike protein, the body knows it's foreign and produce the antibodies that help protect our body from it. If a vaccinated individual now encounters the virus in the future, if you meet somebody who's infected, the body immediately produces these antibodies that bind and neutralize the spike protein and prevent the virus then from attaching to the cells and infect us. So what are the types of COVID-19 vaccines that we have? I'm going to explain it in a second, but just enough to say we either have a killed, a subunit vaccine, a viral vector vaccine, nucleic acid vaccine. We may have heard a lot about the mRNA vaccines, and then what we call virus-like particle. And let me explain them in a diagrammatic representation here. So what are the inactivated vaccines? Simply put, we grow the COVID-19 virus, the SARS-CoV-2 virus, we grow that in large quantities and then kill it chemically. That is similar to, for example, polio vaccine. One of the polio vaccines that we produce it like this, and we inactivate it chemically. So that is just growing the virus and inactivate it chemically. Recombinant protein vaccines or subunit vaccines. We talked about the spike protein. This is how it looks like diagrammatically, this spike protein here. So we can actually either produce this spike protein or produce a small bit of it that is important for the attachment. Remember, we said it attached to receptors. That is what's called receptor binding domain. It's not important for you to know that, but it's important to know that the recombinant proteins is just the spike on its own. We grow it and we add adjuvant and we inject it. You may have heard of what's called the viral vector vaccines. These viral vectors vaccines here, number C, they contain a virus modified to express the S protein. And again, we'll talk about it a little bit. So this is a virus that's harmless to humans. Otherwise, it doesn't cause harm to humans. And then I put in it a small genetic material that then could produce the spike protein when it's injected. And then finally, here's the DNA vaccines and the RNA vaccines and DNA vaccines. This is the Pfizer vaccine and the Moderna vaccines that you heard about, and we'll talk about them in a second. We have this messenger RNA, and we encapsulate it in lipid nanoparticle. The reason why we encapsulate in lipid nanoparticle is that this RNA is very fragile, and therefore we need to protect it. We protect it in these lipid nanoparticles so it doesn't disintegrate before we immunize. That's probably one of the reasons why we store the Pfizer vaccine at very, very low temperature. DNA, it's equivalent to the RNA. It's the genetic material. I put it instead of RNA, DNA. Remember we said we go from DNA to RNA and then from RNA to proteins. So we can in fact inject DNA as a vaccine. There's a company who's doing that. In the human body, it will give us RNA and the RNA will give us the spike protein. Okay, so let's take them one at a time quickly. So the Pfizer and the Moderna vaccine, you can see here is the messenger RNA for the spike protein of coronavirus. As I mentioned, this will be protected in a lipid nanoparticle. When injected into the muscles, it produces the spike protein. So this RNA, as I mentioned, will then cause the production of the spike protein. Once the human body sees that spike protein, it recognizes that it's a foreign material and produces antibodies to it. It then neutralizes the spike protein and prevents then, when exposed to the virus, it prevents it from entering the cells. The second type of vaccines we talked about, and it's important for us, is the Oxford vaccine. If I can go back here, so we heard the Pfizer vaccine has been licensed in Canada and the US and the UK and a few other countries. The Moderna vaccine may be in the next week or a few days will be licensed in the US and potentially in Canada. Canada has actually, and the US have actually pre-purchased some of the Pfizer vaccine and some of the Moderna vaccine, so we're already there. They also purchased the AstraZeneca vaccine and the J&J, Johnson & Johnson vaccines. So for the Oxford vaccine, as an example, as I mentioned, it is a virus that is harmless to humans. It doesn't harm humans. In this case, it's actually a chimpanzee what we call adenovirus. Don't worry about the name. It's actually a virus that infects chimpanzees and actually even then doesn't cause them harm. We take that virus here on the left side here, modify it, and at the same time get the genetic material of the spike protein and insert this genetic material in the harmless virus. So this now virus contains the genetic material of the spike protein. When I then inject it into humans, first the cells express the spike protein and produces some spike protein, and then the body produces antibodies against that spike protein. The next time you see the virus, it does not infect you because you are protected. The last one I want to talk about is the Novavax. You may have not heard about it, but it's also a company, a biotechnology company in the US, that is also producing quite a bit of vaccines called subunit vaccines or recombinant proteins vaccines that we talked about. They are produced by recombinant technology. They are then, once produced, we combine them with what's called an adjuvant that stimulates a strong immune response, and then we inject them. Once we inject them, the body produces, it makes the body produce, in this case, actually, it makes the body produce antibodies to the spike protein, I should say. And then when exposed to the virus, the antibodies will prevent it from entering the cells. This here is a mistake. It should produce antibodies to the spike protein. My apologies for that. So let's talk about the steps we take to ensure that the vaccines are safe. Just to let you know, in order for us to produce any vaccine to license, we first have to go through what's called toxicology studies in animals. We do extensive toxicology in animals to make sure that every organ of the animals is safe after we inject it with the vaccine. We make sure that all the elements of it is safe in animals first. Once we do that and satisfy ourselves that it's safe, we then inject it into a small number of humans, what we call phase 1 clinical trial. Once we show that it's safe in those small numbers, we then, under normal circumstance, we do phase 2, followed by a phase 3. For the COVID vaccine, we actually combine those two, phase 2 and phase 3, and I'll tell you how in a second. So the purpose of the phase 2 clinical trial is to get safety in larger. Remember, phase 1 is small numbers. In phase 2, we get larger number, but we also test the dose, which dose, because we produce the vaccine now, we don't know if you need a little or a lot. So we do what's called dose response in a phase 2. And once we show safety and the dose that we need, we then go to a phase 3, which is scale safety, immunogenicity, which tells us how you respond to the vaccine, and efficacy, how efficacious it is. For the COVID vaccines, that's how one of the reasons why we got them to develop them very fast. We combined phase 2 and phase 3, and you can do that because you can do the dose response first, and once you select the dose, instead of waiting, submitting to the regulators, and then getting their approval to go to a phase 3, the regulators get all of the information on a timely manner. So as soon as we get information from phase 1 or phase 2, we give them to the regulators. So by the time we finish the first part of the phase 2, they already give us clearance to continue on with the phase 3. Importantly to know that the vaccines, by the time they are licensed, they would have been tested in tens of thousands, actually typically 30 to 60,000 volunteers, and demonstrated to be safe and efficacious. So you can imagine before any of us will get dose 1, there will be tens of thousands of individuals have been tested extensively and followed extensively before we get it. Now, once we even get the license to make sure that they are safe, facilities are highly regulated and exceptionally clean. And when I say clean, I'm saying that it's way, way, way cleaner than the operating room where we do, where physicians, surgeons do surgery. Way more clean than that. And then we also do rigorous testing at each step. We don't wait till the end, we test at each step of the process. Now, having said all of that, vaccines may in fact cause some reactogenicity, like for example, injection site pain, redness, fatigue, fever, headaches, and stuff like that. Having said that, that actually means it's working. Remember that you are mounting a very rigorous and vigorous immune response to some foreign entity, and that causes some reactions in your body. When you fall down, it hurts, and part of what happens is that you're recovering because you're getting the machinery of the body to recover your inflammation or pain. It's the same thing with vaccines. I inject you, I get all of the antibodies that we talked about at the injection site. It causes some side reactions. Okay, now, how did they develop the vaccines, the COVID-19 vaccines that quickly? Now, important for us to know that we did not start from scratch. The SARS-CoV-2 is actually the third coronavirus in the past 20 years. I mentioned earlier that there was a SARS-CoV-1 in 2004, 2005. Some of you may remember that. So we knew a little bit about it. In fact, many companies have actually started to develop a vaccine, but the outbreak was finished quickly, so we never had the reason to continue developing the vaccines, but we started. There's also another one called the Middle Eastern Respiratory Syndrome, and that's another coronavirus. Not important, but it's actually started in Saudi Arabia, in camels of Saudi Arabia, and then the camels infected humans, and it became a scare for a period of time. But again, it didn't last long. However, we know a lot then about SARS-CoV-2 because of these two earlier viruses. So we didn't start from scratch. Secondly, the technology has improved significantly. We now know how to produce RNA vaccines and DNA vaccines. At the time of the SARS and the MERS, we didn't. So now we have technologies that once we know the sequence of the spike protein, we can now produce these vaccines very quickly. Accelerated administration, everything was prioritized. So everything that we needed, we got immediately. We stopped, in fact, many of the development of other research activities to focus on the COVID-19 vaccine. Significant funding. Thus far, the world has spent tens of billions of dollars to fund these vaccines. It's currently almost 350 COVID vaccines under development. At least 50 of them already being injected into human beings. Very important is parallel activities. We did research, development, manufacturing all at the same time. As I mentioned, phase 2 and phase 3 earlier when we said clinical testing were done combined. But here, even manufacturing, many of those companies we funded, the organization I consult for started funding and helping people manufacture the vaccines that we will use as soon as we got data from phase 1 trials, which never, ever happens under normal circumstance. We wait until we get a license to manufacture because it's very high risk. Here, we started to manufacture almost all of the vaccines that have been tested in phase 1, all at once, at risk. We know that some of them are failed, and actually some have been delayed, not failed. Some have been delayed. But that we took the risk. Important to know for you is that it's accelerated, prioritized, and concurrent, what we call rolling regulatory review. That is review, the regulators like the FDA and Health Canada started reviewing the data that the companies had as soon as the data is available. Normally, you wait till the end of development, you've done all of your phase 1, 2, 3, and everything else, and manufacturing, then submit to the FDA or other regulators, and they start reviewing, and it takes them months, sometimes years, to give you a license. In this case, they got the information at the same time, almost, that the companies have developed the information. So it's concurrent rolling submissions. As I mentioned, not every vaccine has been successful. They still may be successful, but they got setbacks recently. Sanofi, GSK, and CSL had some setbacks that delayed them significantly. So let me talk about some misinformation that people got. The stuff that I found online and people told me about, although I get a lot almost on a daily basis. So I'll tell you what we know. We don't know everything, but we know a lot so that you can bust those myths. So for example, it says it will make us genetically modified organisms. As I mentioned, the vaccines do not integrate in our genetic material, nor can they. There is no way that even the RNA and DNA vaccines can integrate into the genetic material. And all what they do anyway is to produce spike protein, because that's all what's there in those genetic material. Some things say that they contain technology to link people to artificial intelligence. This is a funny one, interface. Now, in order to do that, they have to have chips, technology chips to do that. Vaccines, as I mentioned, are sterile filtered through very small filters. These filters are thousands of times smaller than the smallest computer chip. And therefore, there is no way I can inject you the chip, because there is no chip that small that can pass through this filter. They did not undergo regular safety check. Otherwise, how did they make them so fast? I think we covered that quite a bit. And we said that we undergo quite a bit of safety reviews. They may contain chips. We talked about that. As I mentioned, it's filtered through what we call a 0.2 micron. Remember, we said the virus is 0.1 micron. So we actually filter all of the vaccine in a 0.2 micron, which is 5,000 times smaller than the smallest chip. So if somebody tells you they inject chips, it is impossible to do that, because we don't have a chip that small, nor can we produce a chip that small. Some people said they contain aluminum nanoparticles that may cause Alzheimer. None of the available vaccines or the ones to be licensed contain any aluminum products. Granted, some of the older vaccines have aluminum as an adjuvant, but none of the COVID vaccines have them. And even those older vaccines that have the aluminum as an adjuvant, the amount of aluminum in them is significantly lower than what you get, than the aluminum you get in food and other things that we consume on a daily basis. So there is no way that this will cause that Alzheimer. Some people say that it causes long-term harm, and I don't even know how they figured that out, because we have not had those vaccines. In fact, the first few people have just been immunized in the UK, none anywhere else. And the vaccines are obviously, as I mentioned, tested at the highest level of safety. The epidemic was created for pharmaceutical companies to make profit. Well, most companies are actually going to provide the vaccine at cost. Both AstraZeneca and J&J said they would produce it at cost, and we know what the price they will charge for it. But even the companies who are making profit, like Pfizer and Moderna, the price of the dose is between $20 and $35, which is significantly less than most of the new vaccines that we have, the Prevnar and Shingrix and Rotarix. All of these vaccines are $150 to $200 a dose. This is 20 to 35. So the companies are not making a lot of vaccines. They are trying to help the population get rid of the pandemic, I should say. Then finally, the question that everybody's saying, should I take the vaccine? And I get asked that a lot. And my simple answer is absolutely. The COVID disease is very severe. And although there are some very promising therapies coming out, like monoclonal antibodies, the best option is still to avoid the infection altogether through a vaccine. Those vaccines, as I mentioned earlier, have been or will be tested in tens of thousands without any severe vaccine-related adverse event. COVID vaccines to date are exceptionally efficacious. In all honesty, when we started this program early in January of this year, I did not imagine that in our wildest dream we get to this number of 95. Just to let you know that the FDA in their guidance to the industry said that they would accept vaccines, COVID vaccines, of 50% efficacy, because that's still a significant efficacy to stop the pandemic. So we are way above that, 70 to 95. The Pfizer and the Moderna vaccine is 95% efficacious. Just so that you know, the flu vaccine, for example, is about 60 to 65% efficacious. So that tells you that how efficacious the vaccines are. The COVID-19 vaccines to date almost completely prevent severe disease. So even those who got infected did not get severe disease. Almost every one of them got only mild disease. So even if you're not fully protected, you at least would not need to go to ICU or get ventilator or something, because none of the people in the trials got severe disease. All current licensed now, or to be licensed, vaccines require 2 shots to 3 to 4 weeks apart. That is true, you need 2 doses. However, the vaccines will provide partial efficacy even after the first shot. So after the first shot, you still get some efficacy from it. And then we have evidence that immunization with COVID vaccines would actually also partially reduce transmission. It does not completely prevent transmission, but if you are immunized and protected, there is a good likelihood that you are not going to be as infectious as somebody else who has not been immunized. And with that, I thank you very much for your attention, and I hope this was informative. And please, if you have questions, as Naguib said, send them to the chat. I'm happy to answer, but please spread the word, make people understand that vaccines are safe and they should get it to help us get rid of this pandemic that we are in. Thank you very much for your attention.
Great. Thank you, Dr. Fahim, for this informative session. Speaking for myself, I know I learned a lot tonight and feel confident that I can make an informed decision when the time comes and the vaccination option is available for me. We have a few minutes for questions. I do have a number of questions and I will read them to you, Dr. Fahim. As they came, I'll not make any changes. So the first question is there one vaccine that would be better or specific to certain population? That is to say, age group or past medical history, etc.?
Yeah. So just to answer the question quickly, the answer is everyone would benefit from the vaccine. Everyone, older or younger. We know now that even the elderly would benefit from the vaccine. So I would not restrict it to any population of any kind. If I understood the question correctly, I would say that it should not be restricted to any kind of population. Everybody would benefit from it.
I think the question is can we make a choice if we have the options? And I'm reading into the question now. Can we make a choice between one vaccine
or the other? That's a very good question. Absolutely. It's a very good question. So obviously with somebody hears of a 90% efficacious vaccine and another one that's 70%, why would I take the 70 and not 95? Now, clearly, if you can get the highest efficacious vaccine, that's great. But I can tell you, if I am offered that 95% efficacious vaccine, but it's not available for another month, I have some another vaccine that's available today that's 70% efficacious. I personally would take the 70% because 2 reasons. Number one, 70% is actually quite efficacious to start with. Secondly, as I mentioned, I know that I'm not going to get severe disease. So while it's not as efficacious as another vaccine, I am not going to suffer the severe disease that caused me to go to ICU and get on a ventilator. So there is no doubt in my mind which one I would take. On the other hand, if I have at the same time the one that's 95 and the one that's 70, clearly I would choose the one that's 95.
Excellent. So we
I hope
have a
that
number of
answers the
questions,
question.
so I'll go through it one by one. Hi, Dr. Raafat. Being mRNA viruses, why Pfizer and Moderna need different temperature? That's question. Also, why Oxford is kept in the normal fridge while it contains genetic material?
Sure. Okay, another very good question. So the difference between the Pfizer and the Moderna, it's what I remember I mentioned that the RNA, we cannot inject the naked RNA. We put in a lipid nanoparticle. So the difference between the Pfizer and Moderna is the constituents of that lipid nanoparticle. They are not the same. So the Moderna one can protect the RNA better in their vaccine than the Pfizer one. And that's why the Pfizer one needs the -70, the Moderna one needs the -20 only. It's the lipid nanoparticle in there that makes a big difference of how to protect it. Now, for the Oxford vaccine, it's in essence a modified virus, a virus vector that we call, and we are able to stabilize those viral vectors. Remember that the genetic material is inside the virus, so what you need to protect is the virus itself, and we are able to protect that virus through formulation, and therefore it doesn't degrade, and we can leave it for about 6 months at 2 to 8 degrees Celsius. I hope that answers the question.
Okay, the following question is continuation. What is the current view about Oxford, knowing that its overall efficacy is 62%?
Sure. So in essence, it's somewhere overall it's somewhere around 60 to 70%. Although some population, the lower dose actually gave a higher efficacy. We don't understand it very well yet. They are having another phase 3 clinical trial in the US that will give us a better indication of what the efficacy is. Having said that, although all of the vaccines, that's the comforting thing, all of the vaccines that we're working on are all against the spike protein. So because we know that they are efficacious with Pfizer and Moderna, we know that you can get them all efficaciously. The AstraZeneca vaccine, on the other hand, AstraZeneca, Oxford University, on the other hand, I didn't want to get into that technical element, but I'm going to go through it very quickly. The spike protein has a certain shape. Once it binds to the cells of the human body, it changes its shape. What we call pre-fusion conformation and post-fusion conformation, before it binds and after it binds. The Pfizer and Moderna vaccine were able to get the messenger RNA exactly right, that when it's reproduced in the human body, when the spike protein is produced in the human body, it forms in a pre-fusion form. The AstraZeneca was not stabilized in the pre-fusion form. So while it's still protective, it's not as protective as the one that I preserved in the pre-fusion form. I hope I didn't get too technical in there, but it's just important for you to know that the shape of the spike protein would have one shape before it attaches to the cell, another shape after it attaches. The Pfizer and Moderna were able to get it in the pre-fusion before it attaches. The AstraZeneca was not stabilized in that pre-fusion form. I hope that was not too technical.
That's good. We have some very informed audience, it seems. So I'll ask you this coming question from the same person, it seems. There is a trial starting to combine AstraZeneca's and Sputnik. How safe and effective is combining two different vaccines?
Yeah. So yes, there is talk. We don't know yet, and I can tell you I'm working with the AstraZeneca folks, and there is at least an idea to combine it with the Sputnik 5, which is the Russian vaccine. It's actually not easy to imagine how the trial is going to be combined, and I'll explain it in a second. But we'll have to wait and see when they come up and tell us how they will combine it. The reason is the Sputnik 5 vaccine from the Gamaleya Institute in Russia actually is two vaccines, not one. The first dose is one vaccine, the second dose is another vaccine. They are both the viral vector, the adenovirus as well, but one is adenovirus 26 and the other one is adenovirus 5, which is a different vaccine. So to manufacture for each human being, I manufacture two vaccines, not one. So when you add the chimp adeno from AstraZeneca, that would be a third one yet. So I don't know how they combine it. Are they going to combine with only one of them, either the first dose or the second dose? It's not clear yet. But sure, combination is something that I can tell you the regulators and the industry will try to do. Instead of getting two mRNA, for example, get mRNA and then a subunit vaccine or a viral vector. We will do that once they're all licensed. I hope that helps.
Just to complete the question, Dr. Fahim, I think the question was how safe and effective is combining the two different vaccines?
So I talked about effective. From a safety perspective, it is exactly the same as the safety of one of them or the other. So it's really no different whatsoever. The safety is the same.
The following question is, would a patient with COVID infection after 15 days of infection, but with lung complications, still be infectious?
So that's an interesting one. The answer, as we know it today, they should not be highly infectious. Although that's why they continue to be isolated, because we are worried that they may still, because the virus may still be there. But under normal circumstances, they should not be as infective as they would be after the first 4 days. But yes, they could still, the virus may still be there ravaging their lungs.
Okay. Another one is, if we have an autoimmune disease, are we more at risk of an allergic reaction?
That's a very specific question. And the answer is, the straightforward answer is, I don't know. But I wouldn't know, on the other hand, why you would have more allergic reaction if you have an autoimmune. I mean, they're in a way a little bit different mechanism. The allergic reaction is a molecule called IgE, and I mean, without having to get into too much detail, the autoimmunity, you'll have autoimmunity against a specific element of self. You're actually, you all of a sudden the body thinks that part of you is not really part of you and starts attacking it. It's a little different than an allergic reaction, so I wouldn't know why, although maybe a clinician can tell us a little bit more.
Okay. How do you explain that 6 people died in the UK from the vaccine?
Not to my knowledge that 6 people died from the vaccine, but people who may have died would have died from other complications, not from the vaccine. To my knowledge, the vaccine didn't cause any death for any person, period, thus far.
Another one for you. When do you think children will be vaccinated?
So the trials that were conducted thus far went down to 16 years of age. They didn't go down below that. That is usually something else we do after licensure. So I can guarantee, although I don't know for a fact, that Pfizer is already, for example, working on lower age than 16 years of age. That's number one. Number two, we know that the severe disease doesn't happen in the young age as much as it happens in the older age. So it was not the priority to start with, number one. Number two, we didn't want to expose the very young population, young and vulnerable population, to a vaccine before we test it in adults who can withstand it. So that's the reason. So, but it will happen.
Okay. The questions are going all over the map, so just bear with me if you don't mind.
Of course.
We heard from some physicians about the adverse effects of having RNA, as it may manipulate the human DNA map and affect human race.
Yeah, that's unfortunate that that you're telling me that physicians would say that. That's just lack of information or misinformation. There is no way that the RNA would integrate itself in our DNA. It is just not possible at all. But even in a very, very, very highly theoretical occasion that it does, all what you will do with that is, first of all, RNA and DNA cannot combine with DNA. So you have to change it into DNA and then combine it. But even if you do that, all what you will do is produce spike protein and therefore get immunity against the spike protein. There's nothing else in the mRNA that we're injecting other than the information to produce spike protein. So it's unfortunate that people who should know say these things. But having said that, hopefully people now are convinced that, at least in this group, are convinced that there is no way you can integrate the RNA into the genetic material.
Okay. As I said, I'm reading the questions as they are,
Sure.
not trying to paraphrase anything. UK now warning that people with medication allergies should avoid the vaccine. Is that true?
Yeah. Yes. So I'm sorry, the internet cut a little bit. Was that the whole thing? That's
the whole thing. That's the whole question.
Yeah. So I mean, again, I'm not a regulator, but I would find this very, very encompassing, way more than it should be. So some people do, we have noticed that there are 3 people specifically, after they started mass immunization in the UK, 3 people got allergic reaction, and they turned out to be allergic to start with. But that doesn't mean that now they encompassed everybody who has any allergic to anything. And they have been criticized in the MHRA, which is a regulator in the UK, have been criticized because that's a very, very wide net. You should not have done that. You should make it very specific. Having said all of that, so if I am allergic to something, I certainly believe that you're still better off getting the vaccine and have beside you an EpiPen or a Benadryl or something, and get the vaccine and benefit from it. That's my view. That's not a clinician's view or a regulatory view. But if I'm given a choice, I say I have the Benadryl and the harm from the disease is way more than the harm from the allergic reaction if I have the remedy, because I know how to treat allergic reaction, anaphylactic reaction. I don't know how to treat COVID-19 yet today. So that's my personal view. That's not the regulatory or a clinician's view. But yes, the UK have done that very wide net. And as I'm saying, they've been criticized by some.
Okay, thank you.